We begin our journey in 1869 with Swiss chemist Friedrich Miescher. He was studying white blood cells, which are key parts of our immune system. Miescher extracted a substance from these cells. It was unlike anything previously known. He called it nuclein because he found it in the cell's nucleus. This nuclein was neither a protein nor a carbohydrate, the two biological molecules known at the time. This discovery was significant. It hinted at the existence of a completely new type of molecule within the cell. Little did Miescher know, he had stumbled upon the blueprint of life itself, DNA. Miescher's discovery was met with skepticism. The scientific community wasn't ready to accept the existence of a molecule as complex and vital as DNA. However, Miescher's work laid the foundation for a revolution in biology. His nuclein would eventually become known as deoxyribonucleic acid, or DNA. This molecule would be recognized as the carrier of genetic information. Following Miescher's groundbreaking discovery, other scientists began to investigate this new molecule. In 1889, German pathologist Richard Altmann coined the term nucleic acid. He recognized the acidic properties of the substance isolated by Miescher. This marked an important step in understanding the chemical nature of DNA. Phoebus Levene, a Russian biochemist, made significant contributions in the early 20th century. He identified the building blocks of DNA, which he called nucleotides. These nucleotides consist of a sugar molecule, a phosphate group, and one of four nitrogenous bases: adenine, thymine, guanine, and cytosine. Levene proposed the tetranucleotide theory, suggesting that DNA was composed of repeating units of these four bases in a fixed sequence. This theory, while inaccurate, stimulated further research into the structure and function of DNA. The mystery of DNA deepened in the 1920s with the work of British bacteriologist Frederick Griffith. He was studying the bacterium responsible for pneumonia. Griffith observed a remarkable phenomenon. He found that a harmless strain of the bacteria could be transformed into a deadly strain. This transformation occurred when the harmless strain was exposed to heat-killed virulent bacteria. This suggested that some transforming principle from the dead bacteria was being incorporated into the harmless strain, making it lethal. This transforming principle intrigued scientists. What was this mysterious substance that could carry genetic information and induce such a dramatic change? The answer came in 1944 with the groundbreaking work of Oswald Avery, Colin MacLeod, and McCarty. They built upon Griffith's findings and conducted a series of elegant experiments. They systematically eliminated various components of the heat-killed bacteria. By doing so, they demonstrated that DNA, and not proteins as previously believed, was the transforming principle. The final nail in the coffin of protein as the hereditary material came in 1952. Alfred Hershey and Chase, two American scientists, conducted their now famous experiments using bacteriophages. Bacteriophages are viruses that infect bacteria. These viruses are essentially packages of genetic material wrapped in a protein coat. Hershey and Chase labeled the protein coat of some phages with radioactive sulfur and the DNA of other phages with radioactive phosphorus. They then allowed these labeled phages to infect bacteria. They found that only the radioactive phosphorus entered the bacterial cells, indicating that DNA was the genetic material being injected by the phages. The Hershey-Chase experiments provided definitive proof that DNA, and not protein, carries the genetic instructions for life. This discovery marked a turning point in biology, solidifying DNA's role as the molecule of heredity. It paved the way for the next chapter in our understanding of the blueprint of life, the race to decipher the structure of DNA.